Hey, hello everybody. Welcome to our blood lab review. Today we're going to cover the general concepts of the blood, knowing what are the major components of the blood and the main characteristics of the blood. You should know that the blood is a type of connective tissue. And as a general rule, connective tissue, you know from AP1 that there's many type of connective tissue, but all the connective tissue will have three major components. We'll have cells, we'll have fibers, and we'll have a matrix. This is sometimes refers as the ground substance. So when we talk about the blood and the lymph are the only fluid connective tissue that we have in our body. You will see that the form elements of the blood refers to the cell component of this connective tissue. Then we're going to see the blood plasma. Within the blood plasma, we have many proteins. The most abundant is called albumin, and that is a type of protein, it's a type of fiber. And then the blood plasma itself, everything else that is not a blood cell or a protein will be the matrix. So the matrix essentially is the plasma without the proteins. In the blood, water represents about 92% of the entire blood plasma. So keep in mind those general concepts, not just for your lab, but majorly for your lecture. Finally, after we review each of the cells, we're going to cover blood typing and blood transfusion. All right, let's talk about the blood in general. When we talk about the blood, the first thing that comes to your mind is probably this red fluid or red substance. Now, have you ever wondered why the blood is red? We're going to try to respond to this question here in this lab to answer this question. But first, you should know that if you take a blood sample from the patient, the vast majority of the time, the place where you're going to take blood is going to be from the vein. You should know that veins are blood vessels that transport blood, but also you should know that we have arteries. Arteries are also blood vessels that transport blood. Now, veins and arteries, they have a different structure, and it's important to know that arteries will carry blood with high oxygen or high concentration of oxygens. Veins normally transport blood with low concentration of oxygen. In our blood, as we mentioned a few minutes ago, we have form elements, we have cells. The cell that transports the oxygen is called the red blood cell. The red blood cell contains a protein called hemoglobin. This hemoglobin is a protein that is able to bind molecules of oxygen, specifically 4 per hemoglobin molecule. And what makes the blood become more bright red or darker red is depending on how much oxygen this hemoglobin is carrying. For example, if one red blood cell is carrying a lot of oxygen, and we said that this hemoglobin is saturated with oxygen, the blood will become bright red. It will be just this red color, which is characteristic of arteries. Arteries, by definition, will carry oxygenated blood. If you have, on the other hand, less saturated hemoglobin or blood with less concentration of oxygen, the blood will become more darker. So the blood that the veins carry, by most of the time, will have this dark appearance. So what makes the blood red is this hemoglobin, is this protein that when it's fully saturated with oxygen, there's a change, a physical change in the color of this protein that makes the blood become red. Other components of the blood is the pH. Remember that the pH is a scale that measure how acid is the blood. You should memorize this because it's important, especially for your lecture. The pH of the blood is between 7.35 and 7.45. This is crucial for the body to maintain these levels, because changes in pH of blood can cause death. So if your pH goes lower than 7.35 is dangerous, or if it goes above 7.45 also can be fatal. Temperature, as you can see here, that the blood is a little bit the temperature is a little bit higher than our regular body temperature, is about 38 Celsius degrees. Viscosity is 5 times more viscous than water, and the amount, depending in males, on average, the amount of blood that we have in the cardiovascular system is about 5 to 6 liters. Females, 4 to 5 liters. Let's talk about the components of the blood in more detail. So we can divide the blood in two major components. The form element, which is composed by the cells. We have three major components here. Red blood cells, which the medical term for those are erythrocytes. Every time you see these sites, refers to the cell. White blood cells, which are called leukocytes, and platelets, which are called thrombocytes. So you should get familiar with all of the three, with the regular name and with the medical term. The second component of the blood, other than the cells, is everything else. That is called the blood plasma. In the blood plasma, we have fibers, which are proteins, like plasma proteins, and regulatory proteins, like hormones, for example. The rest, everything else that is not a protein, that is not a fiber, that is not a cell, is called matrix. And you should know that the matrix is essentially the blood plasma without the proteins. And about 90% of the blood plasma is made by water. Now, when we have the blood, normally after taking the sample from the patient, you're going to place the blood in a tube. That tube is going to be placed in the machine that is called the centrifuge machine. This machine spins the blood very fast, generating a force and making all the heavy and form elements of the blood precipitates or to go down into the tube. Everything else that is more light, or essentially the plasma, will be more in the surface or more superficial. So after centrifugation, that red fluid that we have at the beginning that is still mixed, we can separate it in three major layers, as you can see here. The red one, this yellow, and this thin white coat there. So the yellow part will represent the plasma. It makes sense because 92% of that is just water, so it's not that heavy. You might have some proteins, some hormones, etc. And then it's important that the form cells is, of course, the portion that is deepest in the tube. So here we can see all these red part represent essentially all your red blood cells. This in the laboratory is called the hematocrit. So the hematocrit essentially is the amount of red blood cells. And the white part represents the buffy coat, which essentially is white blood cells plus platelets. Now the hematocrit is important because if you see this sample, for example, we have 100% of this is the fluid. You will see that around pretty close to 50% of the blood is that hematocrit. So it should be the normal value. You can see the normal hematocrit for males is almost 50, it's very close to 50. The same thing for females is very close to 50. If your hematocrit is low, that's called anemia, and if it's too high, is polycythemia, meaning that you have too much red blood cells in your blood. We're going to start with the first form element. This is going to be the red blood cells. The approach that we're going to use is we're going to invest most of the energy to trying to find the more specific characteristics of each individual cell. Remember that in your lab, the goal is that you are able to identify a cell just by looking at it, and just by identify the cell, be able to say also the major function of that specific cell. So red blood cells are going to be the easiest ones because first, they're the red ones. They're the ones that look red. They are the ones that they don't have a nucleus. All the rest of the cells on your body will have a nucleus. It is the only type of cell that do not have a nucleus. That's why when you see under the microscope, sometimes you can see this dark spot in the middle. That is the place where the nucleus be, if those cells will have a nucleus. Now, those by far are the most common form elements of the blood. As you remember, when we looked at the hematocrit, we said that we have the blood, we have the hematocrit here. All these is essentially red blood cells. And then we have a little portion here, it's called the Buffy coat. We'll have the rest of the cells. And then we have the plasma, which 92% of that is water. So that hematocrit will give you an idea that red blood cells are the most abundant types. It's so that you can see here, for example, that the normal value for blood in an adult is between 4.5 and 6 millions red blood cells per microliter. Now, to put you in perspective there, remember that, for example, a tablespoon, it will have around 14 point, let's just around this, 15 milliliters of blood. That is a tablespoon. Right? Now, it will have 15 milliliters. Now, 1 milliliter contains 1,000 microliters. And if you know that just one single milliliter contains 1,000 microliters, see how many red blood cells you have in one single microliter. So in one single microliter, you have 4.5 to 6 millions of red blood cells. That is an insane amount of cells in such a small volume. So we have a lot of them. Other thing is that because the cells do not have nucleus, they are not able to divide. And red blood cells, they have like an expiration day, which is 120 days. After that, red blood cells, they're considered old, and they will be removed by your spleen. So red blood cells are 120. They lack of flexibility, and they will enter the spleen, and there the macrophages of the spleen, which are a specific type of white blood cell, will break down or will destroy all red blood cells. Now let's talk about leukocytes. Now, the term leukocytes is not specific. So when we say leukocytes, we're talking about all these type of white blood cells. So leukocytes, keep in mind that it's like a general term. When we refer to white blood cells, the major function of these cells is defense against disease. Are the cells that fight microbes, and are the cells that protect us. We can classify the leukocyte in two categories depending on the granules in the cytoplasm. When we have a cell, this is the classic representation of a cell. You have the cell, that cell will have a nucleus. I'm going to make an N for the nucleus. And then everything else here is called the cytoplasma. So depending on the type of white blood cells, some of these white blood cells will have granules in the cytoplasma. So we can divide the red blood cells that has granules, those are granular leukocytes. The white blood cells that do not have granules are granular leukocytes. So A for no, and granular, granules. Now let's look at these ones here. So the ones that contains granules, depending on the stain that we use, depending if they react or if they color, we can classify those granular leukocytes in three. Neutrophils, as the name implies, they're neutral, so they do not stain well, and even though they have granules, you will not see a significant color or a changing or a specific color that will attach to those granules. The other two that we have, they will have specific colors: eosinophils and basophils. Eosinophils, most of the time the granules are going to look orange or red. Basophils, blue. The way that I always keep this straight, blue start with V, basophils start with B. If you know that, any other color must be eosinophil. So that is one of the first things that you need to remember. Basophils, the granules will look blue under the microscope. Eosinophils, they will have an orange or red color in the granules. A granular leukocytes, we have monocytes and lymphocytes. Let's review all these leukocytes next. From the leukocytes, you should know that the neutrophils are the most common type of white blood cells in our body. So overall, the most common type of cell in the blood is the red blood cell. But if we talk about the white blood cells, the most common type is neutrophils. Neutrophils are also called polymorphonuclear cells. Poly means many, morpho means shape, and nuclear is the nucleus. This cell has this specific characteristic. It looks like it has multiple nucleus. Poly because multiple, morpho is the shape, and nucleus. So this is very characteristic of these cells. If you see, for example, this is just a cartoon. If you see this in a model where you have a cell that has multiple nucleus, that is a polymorphonuclear or neutrophil. Now, I mentioned a few minutes ago that neutrophils are called neutrophils because they have granules, but they do not stain. So you will not see all these granules that you have here, they will not have a color in a specific. So if you have cells that has granules, but they do not have any color different than the cytoplasma, you're seeing a neutrophil. And the last thing you need to know about the neutrophils: one, that is the more common type of leukocyte. Two, that the nucleus has multiple shapes, like it's multinucleate, looks like it has multiple nucleus. Third, that the granules do not stain. And lastly, when we have a large amount of neutrophils, most of the time it's associated with acute bacterial infection. So those are the four major things that you need to know for your exams: neutrophils, acute bacterial infections. Neutrophils, they have granules, but they do not stain. Polymorphonuclears, they have nucleus that has multiple shapes. And lastly is the most common type of leukocyte in your blood. Now let's look at the eosinophils. Those eosinophils, what you need to look here is the granules. Look at the granules. They have this orange or red color. Um, Eosinophils, they increase in amount in your blood in patients with allergies or parasitic infections, also in autoimmune diseases. Autoimmune disease, parasitic infection, allergies, this is not that common. If you see in a patient, if you take a blood sample of a patient and you get that result that the eosinophils are elevated, those are the major things that you think if that patient might have an allergies or especially these parasitic worms. This is more in the developing countries. Um, yeah, keep in that for your labs and for your exams. Eosinophils will have these colors, these red or orange granules. The last granular leukocyte is the most rare, if you will. It's the one that we have just a few of them. They're called basophils, and if you remember, I told you that they have granules that they will have this dark blue stain. So if you remember blue, basophil, you will be able to say that is a basophil when you see a picture of that. You can see it here in the cartoon or in the model will look like this with those typical granules that they look blue. They elevate those, the count of basophils will elevate also in allergies, parasitic infections, and in some rare cases, hypothyroidism. For your lecture, make sure that you know that this is very characteristic of basophils. They contain histamine, heparin, which is a type of anticoagulant. Now we're going to talk about the agranular leukocytes. Here we have two major cells, lymphocytes and monocytes. Now, the way how you should see a lymphocyte when you look at a picture of that is going to be a cell with a huge nucleus. For example, this cell with a huge nucleus, that is a lymphocyte. This is how it looks in a real blood smear. So you have a cell stained in all these is the nucleus. Most likely, this is a lymphocyte type B. Remember that some of the type B cells are the ones that produce antibodies, right? So antibodies are produced by B cells or plasma cells. Plasma cell is essentially a mature form of a B cell. Most of the time, lymphocytes type B get stimulated by a T cell. Once the B cell gets stimulated, that B cell will start making antibodies, a specific type of antibodies, and the name will change and will become a plasma cell. Again, if you think about antibodies, antibodies are proteins, and it makes sense that lymphocyte type B has this huge nucleus because they contain all the genetic information to make proteins when they are stimulated. They're a little bit bigger than the T cell. T cells in general are a little bit smaller. We're going to see those in the picture in a minute. The last ones are the monocytes. Those are huge. Those are the largest um, cells that we can see in our blood. Interesting here is monocyte and macrophages. So the name change depending on where you see these cells. Now, what I'm trying to say here is that if you have a cell in the blood, it's called monocyte. Now, the monocyte, once this monocyte leave the blood vessel, let's pretend we have a blood vessel here, we have a monocyte here, and you have an infection here. So when a monocyte leaves the blood vessel and enters the tissue, now become a macrophage. Right, so just keep in mind that macrophages and monocytes are the same thing, It's just the name change when they're either in the circulation or when they're not in the circulation. The main characteristic to be able to identify a monocyte is first, is a huge cell, is the largest one, and the nucleus will have these horseshoe-shaped nuclei or nucleus, or if you want, it looks like a U also, right? So this horseshoe shape of the nucleus in a huge cell, that is a monocyte. Monocytes, typically, we can find that elevation of these in blood in people who has chronic infections or chronic disease like tuberculosis, for example. The last form element is the platelets. Keep in mind the platelets are not strictly cells. Instead, they're fragments of cytoplasm of a cell that is called megakaryocytes. The main function of the platelets is hemostasis, is blood clotting. So hemostasis, platelets, blood loss control. For your labs though, you need to know that the platelets are the smallest elements when you look them into the microscope. So in the next PowerPoint slide, I'm going to show you the model that we have in our lab, and we're going to go through all the main characteristics you should look for in order to identify the cells. This is a picture you have in your handouts. So when you look at the cells, your goal for the lab is to be able to recognize the cells just by how they look, and to recognize the main or the functions they have, and when you have a lot of cells or when you have a few of cells in the blood. I would recommend, the easy one is the red blood cells. We have a lot of them, and if you can see in the model, those are the more abundant cells overall. Those are very unique. Always they're going to be red, they're going to look red, and without a nucleus in the center. So all of them, all the red ones here are red blood cells or erythrocytes. Now, the next part is, I will say, platelets. If you use the red blood cell as a landmark, as a reference point, you should know that the smallest structure in a blood smear in the blood are going to be the platelets. So whatever you see as small structures, those are platelets. So all these blue, sometimes they will have different shapes, but your reference here is to localize a structure that is very small. So all these blue ones, or the number 7s you can see here, they represent platelets. So we have now the white blood cells, or leukocytes. And if you remember, we divide the leukocytes in two categories, the ones that has granules and the ones that do not have granules. Let's look at the ones that do not have granules. I think there are going to be a few, and they're easy. The first one are the monocytes or macrophages. Those are the largest cells overall, and you just need to look two things when you try to identify a monocyte or macrophage. One is the size of the cell. If you compare that cell with the rest, they're going to be huge. And the second thing is looking for these U-shaped nucleus or horseshoe nucleus. So we can see here, this is a monocyte, or macrophage is the same. This will be another one, and even this one will be another one. Maybe this is an immature, the nucleus still do not look like a U shape, but this probably is monocyte or macrophage. The next one are going to be the lymphocytes. Now the lymphocytes, the main characteristics of a lymphocyte is that they have a very prominent nucleus. So we have, for example, here, this is a lymphocyte. We have this one is also a lymphocyte. All of these ones that I'm circulating here are lymphocytes. You should remember that lymphocytes normally, the numbers or the concentration of lymphocytes elevate when you have viral infections. So lymphocytes, most of the time they get high when you have viral infections. Platelets, those has to do with coagulation, with hemostasis. Erythrocytes, remember, when you have low numbers, that is anemia. When you have high number of red blood cells, it's called polycythemia. So polycythemia, a lot of red blood cells; anemia, a few red blood cells. Monocyte or macrophages, they tends to elevate. The number gets high when you have chronic infections like tuberculosis, or just in general chronic infections, or when you have fungal infections. Also monocytes, they get high in blood. All right, if we look here, going back to the lymphocytes, you might think, well, these looks a little bit different one another. You should know that the smallest ones probably are T cells. Those are the small lymphocytes, and the B cells are going to be a little bit bigger than the lymphocytes. So we can say that these probably is a B cell, and these probably is a T cell. Recall that the B cells are the cells that produce antibodies. Recall that sometimes when a B cell is stimulated by the T cell, they become plasma cells, and plasma cells is, you can think about a synonym of B cell. They also synthesize antibodies, but a specific type of antibodies depending on the specific infection you have. Then the last portion is going to be those granular leukocytes. So we have here three: neutrophils, eosinophils, and basophils. Remember that overall, the red blood cells are the most abundant cells in the blood, but when we refer to the white blood cells, the most abundant white blood cells in the blood is the neutrophils. And you can see this that is represented here in this model as well. You can see that these are the neutrophils: 1, 2, not this one, 3, 4, 5. So we have a lot of them. The main characteristic is they're going to have granules in the cytoplasm. They will not have a color in the granules. That's why they're called neutrophils, neutral. And they also, a synonym is polymorphonuclears, because the nuclear has multiple shapes. So those are the main characteristics of a neutrophil. Neutrophils elevate when you have acute bacterial infection. All right. The next one, we have two more. We have eosinophils and basophils. I will recommend to memorize basophils. Those are the fewest cells in the blood. They're very rare, but the main characteristic is that the granules are going to be blue. And if you look this one, this is kind of unique, right? And you see the granules are blue. So basophil have these blue granules. They basophils will elevate in allergic reaction. They also intensify inflammation, and also in some parasitic diseases where you have parasites, they can get elevated as well. Finally, we have the eosinophils. Eosinophils will have these granules that sometimes are orange or red. Those are the eosinophils. They elevate in allergic reactions and also in parasitic reactions. Make sure that you do not confuse eosinophils with basophils. Make sure you know the color of those granules. Now, for the rest of the lab, we're going to review the blood type and blood transfusion. So in order to understand that, we should have a better or a general idea of an antibody. What is an antibody, and what is the relationship between an antibody and an antigen? Antibody and antigens are structures that interact each other. Specific antibodies are going to interact with specific antigens. It's important that you know that those antibodies are in the plasma, specifically in the blood plasma. And remember, antibodies are made by B cells. plasma cells. So there's a type, antibodies are type of globulins. They are called immunoglobulins. So now let's finish this review talking about the blood typing and blood transfusion. In this table, you can see the type of blood that we have. Most of the time we use this ABO blood group to determine what is your blood type. You should know that hematologists, there's other type of blood, there are other classifications, they're more specific, more complex, but in general, the vast majority of the time we use the ABO system to determine what type of blood do you have. So in this table, you can see the type of blood, type A, B, AB, O. Now, it's important for this that you understand what are the antibodies and what are the antigens. Antigens always are substances or are proteins that are in the cell membrane of the red blood cells. And if you have a blood type, then the name of the antigen determine what blood type do you have. For example, this is a representation of a red blood cell. This red blood cell, we say that it's a type A blood because the antigens that they have is A antigens, right? A antigen was just the name that they discover when they first studied the blood cells. They found these specific proteins in the cell membranes of the red blood cells, and in order to name those proteins, they assigned letters. So the first one was A, and so all the red blood cells that has this specific type of antigen, they're called A antigen. Therefore, these red blood cells are A blood. The second is that you need to know that in a patient or in a person who has a A blood or A antigen in the red blood cells, in the plasma, they will have antibodies, anti-B. Again, antibodies are specific for antigens. So you can see here that this antibody, anti-B, is specific for antigens B. Therefore, because this patient has this anti-B antibody, this antibody will not react with this antigen, which is A, because they do not match. You can see that in order to for this antibody to react, you will have to have this kind of shape or this specific antigen. So therefore, in a person who has an A blood, they will have in their plasma anti-B antibodies that will not react against themselves, right? So this person will have blood A and will have antibodies anti-B. The next is going to be B blood. They are B because the antigens that they have is B type antigens, and they will have in their plasma, the person will have in their plasma anti-A antibodies. So that means that those are going to react against antigens type A. Because you have B antigens, those antibodies will not react against you. But if you introduce, for example, a red blood cell, like this one here, into the patient blood, these antibodies that are already there are going to react with the antigens and will cause a reaction that is called agglutination, which we're going to review in just a minute. People who has AB blood, they have antigens A and antigens B in the red blood cells. They will not have any antibodies in their plasma. And finally, O type, they have no antigens in the red blood cells, but they have antibodies A and B in their plasma. Another antigen that we use is the Rh antigen. This is also known as antigens D. These antigens D are important because you will see that most of the time when you have a patient and you ask the patient what type of blood the patient has, will tells you A positive, A negative, AB positive, AB negative, etc. So the positive or negative refers to the D antigen, to the rhesus antigen. So this is if the red blood cell, for example, I'm going to make here the representation of red blood cell. Let's say that this red blood cell will have antigens A. So we know that this person has an A blood. And then let's pretend that we have also in the cell membrane antigens D present in the red blood cells. So if the red blood cells have antigens D and antigen A, the person will have a blood A positive. Positive because they have this D antigen. If you have a person with an A blood, which they will have antigens A, but they will not have antigens D, that is an A negative blood. And that is applicable for the rest of the blood types. The way how we test your blood is with an Eldon card. An Eldon card is this that we have here. As you can see, we have in this Eldon card 1, 2, 3, and 4 circles. Each circle will have inside antibodies. So for example, this circle has antibodies against antigens A. This will have antibodies against antigens B. This one will have antibodies against antigens D or RH. And this is just a control. Whenever you put blood, if you put blood from a patient who has A blood, if you put that blood inside of this circle, the antigens are going to react against the antibodies, are going to react against the antigens that person has. So when that occurs, you will have a reaction that is called agglutination, which I'm going to explain in just in a minute. If you put A blood in this circle, you have antibodies B. They will not react against the antigens A because, again, antigens and antibodies, they react specifically with the specific type of antigen. And if you put here in this blood, depending if that red blood cell has D antigens in the surface, they will react. If they do not have D antigens, they will not react. Let's look at this example. Now, I'm going to explain here this is the more detailed way to explain what occurs, but if you want to just be more simple, whatever you see, agglutination is telling you the type of blood. We're going to see examples in just a minute, but I think it's worth it that you at least understand how is that the reaction will occur. So for this, let's look at this example. We have a person who has a blood A positive. Now, we don't know which blood the patient will have. In order to explain this, I'm just going to tell you right away this person has an A blood, right? So you know that the red blood cell of this person will have antigens A plus antigens D, right? When we put red blood cells, or in other words, when we put blood in each circle, what we are going to do is we're going to expose these red blood cells against the antibodies that are sitting in those circles. So for example, if we put blood here in the circle A, we know that there's going to be a reaction because here we have antibodies against the antigens A. Therefore, and you can see the shape of the antibody, once red blood cells gets inside of this circle, a chemical reaction, or these antibodies are going to react against those red blood cells. What is going to happen is that the antibodies will attach or bind with the antigens, and they're going to stick. It's like a glue. It's like a magnets getting attracted to each other. Now, just because you look the shape of the antibody, this antibody has several portions where antigens can fit. So in this case, for example, a single antibody, you can see here, is attached to 3 red blood cells. Once that happens, this process is called agglutination. So all these red blood cells, instead of being floating around freely, they're going to get trapped by these antibodies, and then we can see these with our eyes when we look at the circles, when we look at the reactions. Let's look at these here. For example, if you put blood A in this circle, you will have in this circle antibodies type B. They do not fit. They do not match with this antigen or with this antigen. So you will not have a reaction. You will not have agglutination. And the red blood cells, they're going to be just floating, and the sample will not form agglutination because the antibodies will not react, will not attach red blood cells. The same thing is applicable here. If you have anti-D, for example, these antibodies, because we know that this patient has these antigens here, they are going to react. So the agglutination process is going to occur. Now let's look how this reaction, how you're going to be able to see that reaction once you put the blood there. So it's the same example, the same patient, but you see here the reaction. Whenever you see these clusters of red blood cells, those are agglutinations. Essentially, there's a lot of red blood cells that are stick each other by the antibody that reacts against the antigens of the cell membranes in those red blood cells. You can see here in this one, this is an example of agglutination. You have all these red spots, and see what happens when you don't have a reaction, right? So the easy way to identify what type of blood a patient will have is just essentially just look where the agglutination is. If you see here agglutination in A, you know that that person has an A blood. If you see agglutinate here, for example, there's not agglutination, so the person has not B blood. If you have agglutination antigen D, that means that the patient has a positive blood. So this patient has A positive. Now let's look at some examples, and in your home try to complete those, and we're going to go through them in just a minute. Okay, let's review the first one. So the easy way when you have these is just look the letters, all right? So this one has agglutination. We know that the blood type is A. We know that these are antibodies D, so that means that there's agglutination, there's a positive reaction. That means that person's blood is A positive. Let's look at this example. There's no reaction here, there's no reaction here, but there's a reaction right here. So when we have people or patients who do not have any reactions, that means that the red blood cells, they do not have any antibodies, any antigens in the red blood cells. That means that this person has a blood type O, because if you remember from the picture, our erythrocyte or red blood cell of somebody who has O blood, it means that that person do not have any antigens in the cell membrane of the red blood cells. So that's why they do not react when they are exposed to those antibodies. Now, if you look at the anti-D, there's agglutination, so that means that the person has an O positive blood. Let's look at the next example. We have an A blood, and B is negative, anti-D is negative, so therefore this person has an A negative blood. The next example, you look for the B, that person has a B blood with a positive anti-D, that means B positive. And then the last example here, the person has A blood, B also reacts, so it's AB, and then we have a D, which is positive, the person has AB positive. If you have an AB negative, it will look like this. You have a circle here. You have the 4 circles. So a person with AB negative will look like this. It will have A positive, so it will have agglutination. B will have agglutination, so we know that this person has A blood. And then the antigen D will not react, so it will look in the control will look like this. So a person who has AB negative will look like this. So the only difference here is that the D antigen or the D antibodies will not have any reaction. Let's review the final part, which is blood transfusion. For blood transfusion, we're going to use the same example. And what you need to keep in mind is that because we're going to introduce blood into the patient, you have to be careful to know which antibodies this person will have in their plasma. So in order to explain this, you can try to memorize which bloods are compatible with other type of blood, but really it's something that you can just, if you know the physiology of the antibodies and the antigen, is something that you can just figure it out based on the knowledge that you have. If you're like me, I don't like to memorize the stuff just by memorization. It's better just to comprehend why the reaction will occur. So let's look what happens when you have a patient and when you try to transfuse that patient with different type of blood. Okay, so here I have a representation of a blood vessel. So we have this, for example, let's pretend this is a vein. And this person, if you don't look here, just by looking how the red blood cells look, you know that this person has an A blood. And you see that they don't have any D antigens. In these examples, we're not going to take into account the D antigens, but in real life, you have to account the D antigens as well. But anyway, here you can see this person has an A antigens. They will have in the blood plasma antibodies anti-B. Right? So if you know that, let's look if you can donate blood to that person. So first, we have a donor who has a blood type A. The patient has a blood type A as well. So you see that the red blood cells of the donor are identical to the red blood cells of the patient. So there's not going to be a problem to donate. So this person can donate. I'm going to give you a moment to see if you can do the rest. In the next slide, I'm going to show you the answers. So take a minute and try to deduct if a donor with B blood can donate to this person, a donor AB if they can donate, or a blood O if they can donate. So we can see here that if you have a donor who has a B blood, they have B antigens in the red blood cells. So you can imagine that if you transfuse those red blood cells into the patient blood, the problem with that is when you have the red blood cells that you donate, they have these antigens, but in the patient blood, the patient have those antibodies there. So those antibodies are going to react against the antigens of the donor, and inside of the plate of the patient, there's going to be a problem because all those red blood cells are going to agglutinate. They're going to form essentially blood clots, or many of the red blood cells are going to agglutinate, and that can cause an immune reaction, that can cause a transfusion reaction that in some cases can be fatal. So therefore, you cannot donate B blood to a patient with A blood because that reason. If you have a donor with AB blood, you should know that because they have these B antigens, the antibodies that are in the patient will react against the red blood cells of the donor, so you cannot donate those. Look at the patient with O blood. You should know that the O blood, they do not have any antigens in the surface. So therefore, if you put those red blood cells inside of the patient blood, the antibodies that the patient has will not react against the red blood cells because there's nothing to react with. So in this case, you can donate O or A to this person. Person who has a AB blood, they're called the universal recipients. The reason why they're the universal recipients is because those people, those patients, will not have antibodies in their plasma. So it doesn't matter what type of red blood cells you introduce in the patient blood, there will not be any reactions because all these antigens that those red blood cells had will not react at all because they're not antibodies to react with. So that's why the AB blood is the universal donor. Universal recipient. People who has O blood are considered the universal donor. Why? Because again, if you have a blood type O, you don't have any antigens in the red blood cells. So, for example, let's look at here. What type of blood do you think? Those are four different blood vessels, four different type of blood. Let's look what blood is this one. So the first one you can see is an A blood, B blood, AB blood, and O blood, right? So when you introduce these red blood cells, they will not react with anything because the antibodies that are in the patient's blood, they just do not have anything to attach to the red blood cells. If you see antigens A will not interact with the red blood cells of the donor because there's nothing to interact with. The same thing for AB, they just don't have antibodies. And a person with O blood, they have exactly the same type of red blood cells, so you can donate them. That's why O blood is the universal donor. Now, put attention here that really in this example, we're not considering the Arch-A or D antigen, because if you have, for example, somebody with A positive, with A negative specifically, if you have A negative blood type, they might react with a donor that has an O or A positive RH by the same principle. But for your lab, you just need to know that the universal donor is O and the universal recipient is AB. If you have any questions about this RH, you can send me an email or ask me in class. I'll be glad to show you. But keep in mind that the real universal recipient is AB positive. The real universal donor is the O negative. Let's finish this class just with this example. So let's see if we can figure out which donors can donate to this patient blood. So the first thing that we need to do is determine what is the patient blood. In this case, we see that there's no reaction here. So the patient is not A blood. There's no reaction here. The patient is not B. Therefore, the patient, if it's not A or B, that means that the blood type is O. The second step is to look the anti-D antigen or the RH antigen. In this case, we have a positive reaction, we have agglutination, therefore the blood type is O positive. Now let's look at the donors, what type of blood they have. Donor number 1, we have A, so the patient is A, with a positive D, that is A positive. Donor number 2, there is no reaction at all. That means that the patient has an O blood and negative RH, so it's an O negative. Donor number 3, blood type A, antigen positive, is another A positive. You can see that these and these are identical. The donor number 4, A antigen is negative, B antigen is negative, so we know that it's an O. In this case, we have agglutination in antigen D, that is a O positive. And finally, the donor number 5, we have a positive here in A, so the donor here is A blood, with a negative B, negative D is A negative. Now that we know the bloods, let's look if they can donate or not to the patient. Let's make the patient's blood, right? With the example of the blood vessel. So the blood vessel of a person who has O positive, that means that they have red blood cells who do not have any antigens, but they have a positive RH. That means that they have RH or D antigens in the surface, right? They will have antibodies against A, and they will have antibodies against B. They will not have antibodies against D because it will not make sense, because those antibodies will react against themselves. So it doesn't make sense that they will have antibodies against themselves. So anti-D is negative. They do not have that. But the blood is positive because they have antigens in the red blood cells. Now let's look at here. So donor number 1, they have red blood cells who has antigens A. Just by that, we know that we cannot donate here. We cannot donate this type of blood to this person because the person has antibodies against those antigens. So the first donor cannot donate. Let's look at the second donor here. The second donor here is a O negative. That means that the red blood cells of this donor will look like this. They do not have anything at all. They don't even have any RH antigens. So therefore, these red blood cells can be donated. When you have these red blood cells here, essentially you don't have any antigens to react, so you can donate. So you can donate here at O negative. And remember, the O negative is the universal donor. So essentially, if you see O negative, you can donate to everybody. We see this one, A positive, is the same than the first one. So we know that those two cannot donate to a person with O positive. Then we have donor number 4. It's exactly the same type of blood than the patient, therefore they can donate. And finally, A positive, just by the fact that they have an A antigens in the patient blood contains antibodies against A antigens, they cannot donate. So that is the example here. This is the way that you need to, I recommend that you approach to this, instead of just trying to memorize which blood can donate to a patient. That concludes our blood lab review.